So good evening, everyone. Welcome to the webinar on multi-locus gene sequencing. Um, let's start the webinar. I hope we have a mixed audience here. We have a audience from undergraduates who are learning what is DNA sequencing, and we have a little advanced learner who know how to use various other softwares. Um because I think those are repeated followers of Barcode Biotechnology who are actually offering various teaching and DNA sequencing services. So let's start the webinar. Before we get into it, I want to introduce what is Barcode Biotechnology. They are basically a DNA sequencing company founded 15 years back, and they are most affordable DNA sequencing service provider in India. Those who are in final year UG and PG will know that for basic identification and many more assays, they need to DNA sequence their samples. And these are the machines used in barcode biotechnology for achieving the sequencing reactions. They have a megabase 1000 sequencer, PCR workstations, Mermaid 192X sequence synthesizer. They are actually synthesizes primers, PCR primers, and we have a ABI sequencer. And they offer many various services like bacterial identification service. You can send them an actual sample, live sample, and you can also send them a mixed sample. They will pure culture and identify a bacteria based on 6-ness rRNA sequence, actinobacteria, fungal identification, even cyanobacteria, mushrooms, phages, bacteria infecting viruses. Even they are doing full report services. Like you can send your samples. They are doing all the enzymatic profiling, biochemical analysis, morphological analysis, and even molecular analysis to find what sample it is. You can get all your data. Those are doing PhD can frame a separate chapter from the full report you are deriving because they will provide you pictures and everything of the analysis. Even functional gene sequencing, particular function if you want to decode, Barcode Biotechnology provides such a services. Even multi-drug resistant bacteria, if you want to see a particular multi-drug resistant gene, is it expressing or not, you can use this service. And we of course have a multi-locus gene sequencing, which our today's webinar is all about. We will get into it and we will know what is this multi-locus. There is an upcoming workshop on bioinformatics sequence analysis, how to analyze a DNA sequence. This is mainly because the customers who are actually doing sequencing from barcode biotechnology, when they obtain the machine output file, there are many queries: how to analyze them, how to submit to GenBank, how to draw a phylogram, how to find similarities and variation, many queries. So this is a affordable hands-on workshop because it's just 370 rupees they are offering. It's a 5 days workshop. 5 different softwares are being taught here. So those who are interested can make use of it. And coming to the multi-locus gene sequencing. First of all, why we need a gene sequencing? These days, for detecting any species, any biological species, you need DNA sequences as a proof. Not all genes can be used for a molecular taxonomy. There are few DNA barcodes or molecular markers. Those genes need to be sequenced. In many cases, those are not sufficient enough. So basically, for species identification in taxonomy, it is very important to use the DNA sequence for identifying the species. Why identifying species is such a big deal is in any research, you need to know the species name. You know, taxonomic hierarchy, phylum, class, order, family, genera, and species. We need to know the species name to start any research. For example, you see here, there are more than 3,700 mosquito species belonging to 112 genera, among which only these three genera of mosquitoes, Anopheles, Aedes, and Culex, actually have a mosquito species that can transmit malaria in humans. But among these 430 species of Anopheles, only 40 species, less than 30%, sorry, 10%, only for 10% of the species are actually capable of spreading, can act as a primary vector in spreading the plasmodium. Imagine you are designing a program where you are targeting all the mosquito species. You will collapse the ecosystem because remaining 390 species in the Anopheles is actually doing some ecosystem functioning. They don't spread vectors. They don't act as a mosquito vectors. So it is very important to know the species before devising any environmental plan. Not only in that, say for example, you are into marine conservation. See, there are butterfly-like fishes that are actually corallivores. Okay, they can feed on your corals. For example, keto, dota, and trifascialysis. They can feed on your corals, but these butterfly-like fishes are actually non-corallivores. Example, Heniocus species, they don't feed on corals. Okay, if you are planning a program to target corallivores, it is very much important you know the species before. Even though they are look alike, you need to know how to differentiate them. Okay, even in the medicine, you see, Plasmodium falciparum and Plasmodium vivax both can cause infection, but the treatment method is completely different. For falciparum, for this parasite, they causes severe life-threatening malaria, but they needs artemisinin to treat their, for ACD therapy. And but the Plasmodium vivax actually they can do relapsing malaria, which require primaquine to clear the liver hepatocytes. Okay, so this is very much important that we know the species before we devise the treatment. If you are doing, misidentifying Plasmodium vivax as a Plasmodium falciparum or vice versa could lead to ineffective treatment. You may end up losing life. So diagnosing the species is very much important in various aspects of research, even in the biodiversity research. Always with the conventional biodiversity, you cannot identify the species. Okay, because conventional taxonomy require well-defined key morphological features which are usually present in adult stage of the life forms. Say, for example, in butterfly, definitely species level identification is not possible when they are in larval form. Say, in case of fish fingerlings, you need adult form for identification, but barcoding works or DNA sequence based identification precisely can identify the species. Even in another, there is another problem in conventional taxonomy. For example, in this article, this is my experience during my benthic survey in Bay of Bengal. You see, we actually surveyed polychaetes. These two polychaetes are completely different in their morphology. Okay, even a traditional taxonomist can mistake as these two as a different because they have a different characters here. The morphological character is very different, but to our surprise, the DNA barcodes are 100% same. The DNA sequence between these two species are 100% same. Okay, so it turns out that this species is occurring in oxygen minimum zone, which made them to develop a particular character to absorb more oxygen. So this is actually a morphological adaptation of same species. So see, DNA-based identification can resolve all those things, but you know, um, why multi-locus gene sequencing? So let's understand what is single locus approach. Single locus approach is to identify a particular species, you just sequence a particular gene, you compare it to the database, and then you know what species it is. For example, in bacteria, 16S rRNA can be used as a single locus barcode for a long time now. Is it sufficient enough? Maybe not. You see this example here. I have created this blast analysis. I have subjected 16S rRNA sequence of Bacillus series for blast analysis. Okay, but the 100% similar to Thuringiensis. Again, we have few Bacillus series. Then again, 100% similar to Tropicus. If it is 100% similar to multiple species, how can you derive the species name or how can you conclude what species it is? So that's why multi-locus gene sequencing, because in some species, using single locus may not be effective. Or in other way of saying, the speciation is perfect to recognize them as two different species, but that particular barcode marker where we are using to identify them may not be evolved enough. Another example is here. This is the 16S rRNA of E. coli. You see, they are 99.24% similar to Sigilla. You see, if you have the cutoff as 3%, definitely they are not falling. You see, even there are 5 genera inside the cutoff of 3%. You see, till 97 point something, the cutoff is there. So definitely this 16S rRNA is not working. But if I substitute the 16S rRNA gene with RPOB, you got all E. coli identified. Okay, same species, 100%. So that's why. So can we substitute RPOB for all the species? Maybe not. Why? Because you, for DNA-based approach to work, you need to have an enriched database all the sequences of RPOB of all the species. Maybe I don't know, 5,000, 5 to 7,000 of bacterial species are documented. You need to have those sequences already. It's a costly affair. So multi-locus gene sequencing. For example, in Candida, it is well-known fact that to resolve Candida albicans complex, they use both ITS gene, internal transcript spacer, and MSPI, ALUI, MSPI. So various other genes, including GPI, can be used along with ITS for better resolution. Okay. So let's see. There is another concept called multi-locus sequence typing. What is that? You remember during your pandemic time, whenever a SARS-CoV-2 evolve, they will be naming something like this: alpha B.1.7 or beta 1.351. So what is these numbers and how are they named? Let's understand that. For example, I want to do a multi-locus gene sequencing for precise identification of Neisseria meningitis. Okay, so if I choose these 7 genes that is being located here, okay, to identify that, maybe ABCZ gene, maybe they are in 7 copy numbers. 7 copies may be present in the database. Each copy number, if they are similar, they are given one allele number. Okay, say for example, if 7 copy number has a one nucleotide at least different, then 7 numbers of this particular gene will be recognized as allele 1, allele 2, allele 3, allele 4, and allele 7. So when I do a blast analysis, if my sequence match perfectly with allele 7, then I will write the allele number as 7 here. Here it is perfectly matched with allele 3. Okay. But if it, when I say perfectly matched, it is 100% matching. Okay. If it is matched only 99.5% and only one nucleotide is variable, then I will give a new allele number for my sequence, which is 8. Because only 7 sequence is present in NCBI, now it is 8. Likewise, here it is given as 42. So when this is something to create an allele profile. Say this allele profile of Neisseria meningitis is 342.75468. How it is possible? All these genes are arranged in this manner, and during the blast analysis, they are checking for 100% similarity to take the same allele number. If it is one nucleotide different, then the allele number, we can assign a new allele number and new profile is created. This is just to keep the track of pathogens. This is very much practiced in the clinical settings. Okay. So basically, multi-locus gene sequencing is usage of multiple genes for precise identification. We have just seen various examples of why to use such a multiple genes. Okay. And see what will happen if you use a multiple genes. When you draw a phylogram using single gene, see the resolution of the phylogram. But when you draw a multiple genes, see the resolution of this phylogram, that is combination or concatenation of genes. What is this concatenation? Say, for example, to identify your bacteria, you sequence 6' rRNA and RPOB. So here is the 6' rRNA gene 1 and RPOB gene 2. The last base pair of the 6' RNA will be followed by the first base pair in the RPOB. You simply copy your sequence and merge it as a one sequence. Okay. After merging all the sequences, multiple genes you are using to precise identification, now the resolution of phylogram will be much more better than previous. Okay, using single gene. That's a entire idea of using multi-locus gene sequencing. Okay. See here, we initially saw for Bacillus how Thuringiensis and series are, they are overlapped in the blast analysis. But see here, when we use a multi-locus gene sequencing, this is for those who are listening to this for first time. This is a phylogram. You are supposed to see from the left-hand side to right-hand side. Okay, so there are 2 branches, then 2 branches become 2 more branches. Likewise, there are further subdivisions. All right. So when the sequence are branched in the same branch, meaning they are more similar. For example, here Bacillus series branched with all other species, so that they may all belong to same species. Here Bacillus series are kept, or Bacillus mycoids are kept away from the Bacillus series. If you want to reach Bacillus mycoid, you need to track along this and then go and reach the Bacillus mycoids. So they are kept away, meaning they have more variation. when compared to the DNA sequence of baseless series. So phylograms are graphical representation of similarities and variation among the DNA sequence. So when we add these three genes, this is actually a title of the paper, you can check it out. Usage of 16S rRNA, 23S rRNA, gyrase B gene, sequence based to determine the phylogenetic relationship baseless series group. So these closely related baseless series, baseless series like species like Bacillus thuringiensis, mycoids, anthracis, they can be clearly resolved based on these three gene phylogeny. Okay. Now let's see what is happening in the lab, how this multiple locus gene sequencing is done. Okay. Very simple. Whenever you send a sample, they will go for a DNA extraction. Okay. With the extracted DNA as a template, whatever genes they are choosing for multiple, from the multiple locus, each gene will be considered for sequencing. Each gene will be conducted with, individual PCR reactions having a target primers in them. In many cases, even multiplex PCR is also possible, where mixture of primers will be used for targeting, especially when the amplicon size is differential, that can be possible. Otherwise, individual reactions are carried out in Barcode Biotechnology for Multilocus gene sequencing. They are doing individual PCR reaction. These PCRs are actually split into two samples for forward and reverse sequencing. Here they have mini ion, but we are using Lemna and Sanger sequencing for sequencing. So once you got for every sample, if you are choosing 5 genes, then there will be 10 reactions, 10 PCR reactions at the final step, single standard PCR. Then 10 machine output file is generated, and forward reverse sequences are compiled to, create one single sequence, and then concatenation will happen. They will merge the sequence from first gene with second gene, followed by third, fourth, fifth. They will merge the sequence and they will use the sequence for phylogenetic analysis. In Barcode Biotechnology, they are providing pairwise distance data and phylogenetic analysis as a part of the service of multiple gene sequencing. Okay. Again, here is another example. In resolving the phylogeny of a lizard species, you see before the multi-gene, multi-locus gene sequencing or single locus sequencing, or what will happen with the multi-locus gene sequencing? Better resolution and better species identification. Okay. So this webinar will be a very short webinar. So I want to conclude it with what are the challenges of multi-locus gene sequencing. Yes, since you are treating your sample into 5 PCR reaction, 10 single standard PCRs, and then machine output file, it will be time consuming. It costs us also every gene. Still, I think Barcode Biotechnology is India's most affordable DNA sequencing service, but still every gene costs us. Gene bias selection, you should be very clear about, say, for example, in bacteria, better go for RPOB and 16S rRNA, not with amylase, gene and 16S rRNA. You should know what gene to select for multi-locus gene sequencing. And it requires complex data analysis, but I don't think it's very complex. Barcode Biotechnology can resolve that. That's why they conduct these workshops and webinars. Future perspective, I think whole genome sequencing will give you the better perspective, but still it is very costly when compared to multiple gene sequencing. Okay, multiple gene sequencing is at least 2 times that of single locus sequencing, but whole genome sequencing costs you 12 times more that of single locus sequencing. Okay, so cost-wise, it is much more better for species identification. Okay, so this is a service I was talking about from Barcode Biotechnology. They are giving you can identify precisely the bacteria using multiple genes instead of just one single gene. And this is a hands-on training we are launching this time. It is very affordable training. You can do it from your home, just 1 hour every day. Live sessions will happen, and even recorded sessions will be circulated in the group every day after the live session. If you cannot make it for their particular day, this is going to happen between 7th and 11th August. This is for UG PG students who are initiating their projects, even PhD students who want to know how to analyze the DNA sequence, postdoc scholars having issues in DNA sequence analysis, Sanger DNA sequence analysis, even faculty members who want to learn for their research team DNA sequence analysis can use these services. Okay, and okay, this brochure will be circulated today to even through your email. You can receive this brochure. We'll see. These are various other services provided by Barcode Biotechnology. Not only based on bacteria or microorganism, even animal identification surveys, insect identification, community samples can also be barcoded for meta-barcoding. We can extract DNA and identify the plant tissue samples, herbal samples, even forensic samples. You can simply send us the fecal samples. We can decode from which host the fecal sample is collected, or what is the species name, and even dietary composition of the feces can be decoded. Even the birds or other animals that are restricted to be sampled, you cannot catch them for sampling, but their dried fecal material can be used to decode what species it is. Very much possible. Even simply collecting air samples through a filter membrane, we can extract DNA from the membrane and decode the entire biodiversity collected in the air. Okay. So Barcode Biotechnology provides various other services, and Barcode Academics is a small unit in Barcode Biotechnology into various webinars and workshops. These are the forthcoming workshops. You will receive them through your email or through your WhatsApp. Okay. There is a free workshops on usage of AI in phylogenetic construction, and there is a free workshops on PCR primer designing. And we have this. This is a little paid workshop, but still very small money for 5 days workshop on DNA sequence analysis, especially for one who wants to learn these tools: how to do blast analysis, how to use a DNA sequence to find a species name, how to compile forward reverse primers using bioedit software, how to use the clusterlex for multiple sequence alignment, how to use Mega for pairwise distance data and creating a phylogram. For those who want to learn such a thing and use it for their use it in your project, UG or PG project, or even in PhDs and postdoc work. This workshop is a base for that. As I said, there will be it is outside of academic hour, and a pre-recorded video will be shared every day, so you can have your own time in analyzing it. We also have a free webinar coming on fungal species identification very soon. We'll announce that with the brochures. And we are actually planning 3-month research project in DNA barcoding. Those who are interested, those who have to find various other institutions for the barcoding project, they can use this opportunity. We are going to give 7 free DNA sequencing for those students who are taking up this project. It is a 3 months project, probably starting from November to January. And Barcode Biotechnology will guide you in, we will frame a project, and it will be DNA sequence based project. The certification will be done, and the support in thesis preparation, even a manuscript. Many of those who are aspiring to go for European country, for various international fellowships, having a research publication become a mandate. So we were working on various other aspects of how to make a research publication out of it. And planning a publication in short span of time like 21 days or a month is a really cruel thing. We need at least 90 days to see and plan for a publication. That's why this research project is planned, and those who are very much interested in DNA sequence based research should utilize this opportunity. Okay, for all those things we will find a brochure. It will be circulated very soon. And thank you for joining today's webinar on multi-locus gene sequencing. And I hope this was a little useful for you for your research. And for regarding the e-certificate, a link will be shared right now in the comment section of the YouTube link. It will be active for 1 more hour. Please fill that Google form to receive your e-certificate. E-certificates will be provided before 9 p.m. tomorrow. You will receive it through an email. Okay. So thank you for joining us. Happy barcoding.